Assalamu alaikum first year. Now the lectures are the chemistry of nucleotides. This is my first lecture on chemistry of nucleotides. Not only they are the precursors of nucleic acids, they are involved in the formation of nucleic acids, as nucleic acids contain two types of nitrogenous bases, purine and pyrimidanes. These nucleic acids, they may be a RNA or DNA, and they contains the purine and pyrimidanes. These nitrogenous bases or nucleotides or nucleosides, they are involved in many metabolic functions in our cells, in our body. They are the they are involved in the energy metabolism as energy, as in as a energy currency, as in the form of ATP, GTP, and they are the actual gives free energy to do a work, and they are also involved in the regulation of enzyme activity. The enzymes may be equivalent. There is a regulation, covalent regulation, as maybe a phosphorylated or dephosphorylated, and all of these phosphorylation has been done through these nucleotide for a nucleoside triphosphate, such as ATP or GTP, and they are also involved in the signal transduction as a second messenger, like cyclic GMP and cyclic AMP. So they are involved in the signal transduction reactions, signal transduction. During the signal transduction, the ATP, it is transformed into a cyclic AMP, and the GTP, it is transformed into a cyclic GMP. So they are also involved in the signal transduction reaction, and they are also involved in the structure of many coenzymes. They are the with the coenzymes, they are formed by the active vitamin, from the active vitamins, and many of the coenzymes, they contain the nucleotides, and they are similarly, they are the principal donors and acceptors of the phosphoryl groups in the metabolisms. They are involved in the phosphorylation reactions during the metabolisms. As I have already told you that they are involved in the covalent modification or of the enzymes, as phosphorylated enzymes or dephosphorylated enzymes, and all of these, they have done through the ATP or ADP. So these not only these purine and pyrimidine nitrogenous bases in combination with the phosphate or ribose to form nucleotides or nucleosides, they are not involved in nucleic acid, but they participate in many metabolic reactions. Like nucleoside triphosphate or diphosphate, like ATP, ADP, they are the energy transduction reactions. If high ATP ADP ratio present in the cell, it means the cell is at high energy state, and all those metabolic pathways which give the ATP, they are inhibited, and all those metabolic pathways that utilize these ATPs, they are activated. So the presence of ATP ADP ratio is very important for the cell, for those metabolic reactions which may be activated, or those metabolic reactions which may be inhibited, and they are also the this ATP ADP ratio also involved in the oxidative phosphorylation. High energy state mean that there is no need to form a ATP. So those metabolic reactions which ultimately form the ATP, they are inhibited. These are also, there are the derivatives like sugar derivatives like UDP glucose or UDP galactose. These are the sugar derivatives. Here the sugars, they are combined with the uridine diphosphate or galactose combined with the uridine diphosphate to form the sugar derivatives, and they are very much important during the biosynthesis of starch and glycogen, and also they participate in the sugar interconversions lactose metabolism. Here the UDP glucose and UDP galactose, they are interconvertible, and similarly during the biosynthesis of glycogen, as the glycogen is the storage product in the humans, so they are also involved. Just like a UDP glucose, it is the active form of glucose which is involved in the glycogen biosynthesis. So these sugar derivatives, they are very much important for the biosynthesis of starch as well as the glycogen. Similarly, another derivative which is known as the CTP acyl glycerol, and it is also the intermediate in the lipid biosynthesis. Phospholipids, they are synthesized mostly through their CTP acyl glycerol. So CTP acyl glycerol is another derivative of nucleotides which is involved in the lipid biosynthesis. Similarly, ATP dependent phosphorylation of the key metabolite, as I have already told you, that they are involved in the activity covalent modification of enzymes, which also depend upon the ATP ADP ratio, and these are also helpful in the allosteric regulation of the enzymes. If high energy state, then they will inhibit those metabolic processes that ultimately yield the ATP. Catabolic processes or anabolic processes, they depend upon the ATP ADP ratio in the cell. Cyclic AMP, cyclic GMP, they serve as a second messenger, and in the during the hormonal regulated events. Similarly, they are also important in the medical applications, as they are the analogs, purine and pyrimidine analogs, which are utilized during the chemotherapy of the cancer, AIDS, and also they are the suppressors of the immune response during the organ transplantation. So medically they are also important. They are basically the derivatives of halogen thiols or additional nitrogen atoms, and they form the analogs. They are not true purine or pyrimidine, but they form the analogs which are helpful during all of these treatments. They are utilized during the chemotherapy of the cancer, or maybe AIDS, or they maybe suppress the immune response. These are the basic structure which is present in the adenosine monophosphate AMP. Here it is, this basic structure is present in most of the coenzymes, which is used in many metabolic pathways. This R double prime group, R prime group, it differ, or R double prime, R prime. These groups, they are different in many coenzymes, but they are similar. Their basic structure is similar in most of the coenzymes. Like this is the table which show that how R prime, for example, active methionine, it contains hydrogen at R prime position, R double prime position. It also contains the hydrogen, and similarly amino acid adenylates, it contains hydrogen and hydrogen at R prime and R double prime position, but at R prime R position it contains the amino acids. Similarly, active sulfate 3 prime 5 prime cyclic AMP, NAD, NADP, FAD coenzymes, all of these coenzymes, they are involved in different coenzymes. The basic structure is same, but there is a difference at these positions, and so they perform their functions. This is the structure of the cyclic AMP. Here it is formed from the ATP. This is the 3' to 5' cyclic AMP adenine. In this case, adenine, it is the nitrogenous base, and similarly this is the structure of cyclic GMP. Here the guanine is the structure cyclic GMP. Cyclic AMP and cyclic GMP, all of these two structures, they are very much important during the signal transduction, and they are also, in this case, the nucleotides involved in these structures. Similarly, another structure which is known as the S-adenosyl methionine. This S-adenosyl methionine, it is the methyl donor, active methyl donor. Here the methionine, this one is the amino acid, and this labile methyl group has been donated to different compounds methylation. For example, even the methylation of DNA take place at certain region. This methylation, it is the involved in the chromatin remodeling, as transformation of the euchromatin to heterochromatin, which is inactive gene. And similarly, this is the, basically it is a nucleotide. Here the adenine is the nitrogenous base, then sugar residue, and then it is attached to the this amino acid. So it is the derivative of nucleotides, and it is involved in the donation of the methyl group to different compounds to perform or to become the chemical reaction. Another compound known as the adenosine 3-phosphate 5-phosphosulfate. This is another active compound, and this active compound, it is the here the 3' position also contain the phosphate residue, and the 5' position also contain the sulfate. This one, this activated sulfate, it has been given to the different compounds like the sulfur glycosaminoglycans, GAGs, sulfated GAGs. They also use this compound so as to sulfate for the sulfation. So it is very important, and all of these, they contain the like adenine, ribose, and phosphate nucleotide. Nucleotide, it is the component of this compound. So nucleotides and nucleosides not only involved in the DNA RNA synthesis, but various metabolic reactions, various cellular intermediates, and various their concentration also inhibit or activate various metabolic reactions. Thank you very much.